Hello, and thank you for attending this session entitled RDCA-DAP Facilitating Collaboration in the Pre-Competitive Space, Case Study in Friedreich's Ataxia. My name is Megan Cala, and I'm a postdoc of Pharmacometrics on the quantitative medicine team here at c-path. And I'll now turn it over to my co-presenter, Christian Rummey, for his introduction.
Thank you, Megan. Christian Rummey, I'm a scientist and statistician, and my story with FA started more than a decade ago when I was involved in one of the first phase 3 programs that were run in the disease, a compound called idebenone. And then really I have never left the space ever since, and for the last couple of years been a statistician on one of the 2 large natural history studies that are available for Friedreich's ataxia. And the work I've been doing there is of course funded by FARA. And we're going to do this a little bit back and forth, so Megan will introduce you to the platform and what we're planning to do with it. And I will speak about the disease and the endpoints and how to measure really FA. So what is FA? It's a life-shortening degenerative neuromuscular disorder. It affects about 1 in 40,000 people, and it's easier to remember if you say there's about 5,000 patients in the US and about 10,000 supposed to be in Europe. It's the most common hereditary ataxia. Its inheritance is autosomal recessive. So carriers are usually unaffected or asymptomatic, and children have a chance of 25% to inherit the disease. The genetic cause is a repeat expansion in the frataxin gene. And this expansion usually has lengths up to 30, and that's the normal length. But in FA patients, it goes up to more than 1,000, sometimes 1,700 repeats. And these repeats cause a frataxin protein deficiency, leading, among other things, to iron accumulation in the cells, which then leads to cell death. And it's important to notice that among the neurological endpoints that we'll mainly discuss today, FA patients also suffer from cardiomyopathy, which is the leading cause of death. It's just that the development of endpoints in the cardiac phenotype has turned out to be much more complicated and is not as advanced as on the neurologic phenotype. And since it's a multi-system disorder, it affects also other symptoms, leading to an increased number of diabetes. So this genetic defect, the number of GA repeats, you can correlate with the age of onset of a patient. So on the y-axis, you can see more repeats lead to earlier age of onset on the x-axis. And this correlation works pretty well if you look at this whole natural history scale. However, it's slightly modified towards earlier age of onset. And if you look at the red group, then you see that the correlation doesn't really work anymore in really young patients. So the disease progression in FA starts with diagnosis, and that at diagnosis, the patient is usually asked, when did you perceive your first symptoms? And that happens typically about 2 to 4 years before diagnosis. And then after diagnosis comes a period of loss of walking capacity that then leads to use of walking aids and eventually loss of ambulation. And after that, people become full-time wheelchair users. Now, it's important to note that many of the measures that are used today focus on walking and balance and gait, but about 50% of the overall population of people living with FA is actually non-ambulatory. So it's very important to also develop measures on this side. Now, I've mentioned age of onset. I have dashed lines here because age of onset can really occur at any time in life. It happens in really young children, and as you've seen in the previous slide, also during adulthood. And I've tried to visualize that here by showing age of onset as a circle and age of diagnosis as a blue dot. So there's really early patients, and I've shown here up to 30, but in the database we have people with onset up to 70 years of age. And you can see easily if I put the first milestone, loss of ambulation, on the scale, then you see that this can happen very early, in basically early teenagers, in the most severely affected, while people who have an adult onset basically can walk for decades after onset. Now, when you think about the measures and the development of the disease, it's clear that the younger affected patients are at their onset, the more this will affect with their neuronal development. And there are several thresholds that have emerged that are somewhat separating the clinical presentation of these patients. And certain categorizations have been proposed, like everyone who has an onset earlier than 8 years is early onset, and then we have typical, intermediate, and late onset. So there's a huge diversity. That's the point I'm trying to make here. If you look at the distribution, most patients are actually diagnosed between 8 and 14 years. But of course, there is a huge selection bias in this graph, as of course, the milder patients are, the less motivations they have to actually participate in such a study. Now, the next slide, I want to talk about the clinical trials that have been collected in the database at c-path. So we have the early studies on idebenone that I mentioned before, and then we have 4 other clinical trials. And because the disease is so diverse and patients can behave so differently at different ages, it's important to contextualize and have the background of these studies in mind when you analyze them. And yeah, so that was the introduction to the data and the disease. And now I'm going to hand it over to Megan to speak about the platform that c-path has built around it.
Thank you, Christian. So yes, the FAICD is hosted on RDCA-DAP, and it can be accessed by logging into FAIR Data Services. And you can select the FA aggregated data to get some more information about its contents, as shown in this screenshot here. And this will include some relevant descriptions related to the catalog info, keywords, data dictionary, as well as field level metadata. And also from this page, if data access request workflows are enabled, users can request access to this data set, and it will be delivered to your workspace. The next slide, please. And so to give you an idea on the size and contents of this data set, as Christian discussed, there are 6 clinical trials and 1 long-term natural history study, which amounts to approximately 1,500 subjects and close to 5,500 observations. So this is a very rich database. And the 2 plots on the bottom left show a breakdown of subjects per study, as well as the average number of observations per subject by study. And then to the right, we've summarized the categorical and continuous covariates of interest. These include race, ethnicity, sex, and baseline ambulation status. And for continuous covariates, we have age, baseline age, age of onset, and the 2 GAA repeat lengths that Christian discussed. And these are all candidate covariates that we can incorporate into our analysis as we try to understand the variability in the baseline severity and the progression rate of FA. Next slide, please. And to further interrogate the FAICD, I want to highlight the work of Kurt Michaels. He's a member of c-path's data science team, and he has developed an R-Shiny GUI that's available as one of the RDCA-DAP's mini apps. And this FA interrogator can be accessed under the mini apps tab and launched directly from the workspace. And within this app, you can perform all sorts of data exploration and data visualizations and statistical summaries in a very simple and user-friendly manner. And in this screenshot here, you can see a histogram and a normality check of one of the available endpoints from this data set. And all of this can be executed without having to type a single line of code. And within this app, you can also filter through particular dataset domains to generate subsets for analysis. And this was one of the RDCA-DAP's pilot projects, and it was covered in much greater detail during one of the webinars that was hosted earlier this year and can be accessed on c-path's YouTube channel. And additional analyses can also be executed directly within the Workspaces environment using available open source software. So there are mini apps available for both RStudio and Jupyter Notebook so that users can utilize both R and Python respectively via these mini apps. And here I'm showing you an example analysis that I ran of the baseline age across the available studies using the RStudio mini app. And what's really convenient about working within the platform Workspaces is that you can easily share scripts and figures and files instantaneously with your collaborators. And if we take a closer look here at these baseline age distributions, we can see that the median age is slightly variable from study to study, ranging from the low teens to late 20s. And as you can see, the range of these included ages also varies from study to study. And this is due to the different enrollment criteria that correspond to each of these studies. And this is important to note because what this also means is the underlying disease severity of these included subjects is also going to vary. And in addition to age, we can also examine the ambulation status of individuals at baseline. And as Christian discussed earlier, the loss of ambulation is an important milestone in the progression of FA and can therefore indicate the diversity in terms of disease severity among patients at baseline. And this figure shows a histogram of the baseline ages of all patients across the entire FA-ICD. And by stratifying by ambulation status, where blue indicates the ambulatory individuals and orange identifies the non-ambulatory, we can see that at baseline, there are many patients that have already reached that non-ambulatory milestone. And if we compare the area, the blue area versus the orange area, as you move from left to right, we can see that the proportion of ambulatory individuals decreases with increasing age. And in this next visualization, we've separated the ambulatory individuals on the top from the non-ambulatory individuals on the bottom. And this figure includes all observations, so not just the baseline visits. And now the colors indicate the 4 different bins for the age of onset. And what you'll notice is that the ambulatory section, there is very little red and blue past the age of about 30. And that's because those earlier onset groups typically progress faster. So loss of ambulation occurs sooner in those individuals. And overall from this graphic, we can identify the underlying variability with respect to disease severity in these various age of onset groups or age-based populations, which is often the case in clinical trials. And now back to Christian to discuss the various endpoints in FA.
Yeah, so Friedreich's ataxia is usually measured and assessed with neurological exam scales, rating scales. And one of the 2 most important ones is the mFARS, the Modified Friedreich's Ataxia Rating Scale. It's a multi-component scale that covers bulbar function and then upper and lower limbs, as well as balance and walking functions. That's the upright stability score, which is arguably the most important one, important scale in ambulatory people. Then in the gray box in the middle, you have a functional disability staging that assesses the disease on a broader scale and focuses on milestones. And we have an activities of daily living score, which is a patient-reported outcome. And lastly, we have also direct functional measures in the database. These are mainly assessing walking and upper limb function, and the two most important ones are the time 25 foot walk and the 9-hole peg test. It's important to know that there's quite a few additional measures in development. Just these are the two that we have vast amounts of data of. Now, if you put these measures in the context of my disease progression slide here, you can see that the FARS was actually designed to cover the whole disease range. But if you look down at the subscores, you will see that, of course, certain scales only work in certain disease phases, and balance and walking only works in ambulatory people. And we could show that, for example, upper limb function really is only, or is mostly lost only in the non-ambulatory phase of the disease. And the same is true for direct functional measures, where we have the time 25 foot walk, obviously in ambulatory people, and the 9-hole pegboard test, which is actually the only outcome that seems to work throughout the whole range at the same quality. And with a PRO, it's a little bit the same like with the rating scales, because they do work over the whole range, but they behave differently when people start to bother about different walking functions. And yeah, that's it. So now I'm going to turn it back to Megan to show you the amount of data we have in the database.
Thanks, Christian. And so as for the endpoints available within the FAICD, this figure summarizes the number of unique subjects contributed by the various studies that are available at baseline for each of the endpoints. And all of the endpoints that Christian just discussed are included along the x-axis. So as you can see, the studies vary slightly with respect to which endpoints are included. And the last two bars on the right show combinations of endpoints. So if we were to consider multiple endpoints in our analysis, understanding how that may alter the number of patients and data points that we have to develop our models from. And on the next slide, for the endpoints that are composite scores, such as mFARS, which I'm showing on this slide, we can break those scores down to their item levels. So in the case of mFARS, there are 18 items. So those are the 18 bars shown here. And for each item, we can note the distribution of scores within the population. And you can read this figure like a heat map, where the darker regions correspond to higher scores or higher severity. And what this figure is conveying is the severity of the population at baseline per item. So for example, the lower limb subscore C, and that's the third subscore from the left, the population in this data set spans the severity spectrum. So you have individuals that fall into each of these 5 shades of orange. And compare that to some of the bars in the upright stability or subscore E items on the far right, where some items, the majority of the population is already at the max severity at baseline. And this is particularly important when we're looking at these scales longitudinally, because subjects that have reached the max at baseline can artificially show slower progression or no progression at all. And next slide, please. So what we've discussed today here is really an overview of the types of analyses and of these clinical trial data sets and natural history study that are very important and align our thinking in the development of disease progression models. And these are mathematical models that describe and quantify disease endpoints and use available information in the data sets to identify sources of variability in the patient population. And this is really the current status of this ongoing effort. And as we continue on with next steps, we can also add on a placebo effect model if we can confirm that the data will support this additional model component. And then we can simulate asymptomatic drug effect and use the models to estimate the sample sizes that would be required to detect a significant effect. And lastly, on the final slide, I will end here with the big picture of this effort, which is a clinical trial simulation tool that we aim to develop to be hosted on the RDCA-DAP platform. And with the intended use for this tool being to inform future clinical trials in FA by means of calculating sample sizes, optimizing trial durations and treatment effect times, and answering trial design and analysis questions to support the greater FA community. And with that, we would like to thank everyone for your time here today and attendance, and use the remaining time to answer any questions that have come in. Thank you.
Hi again. So I think I will kick off the Q&A with just a clarifying comment here. So a question came in that what is meant by baseline. So baseline for these is not necessarily time at diagnosis or time at symptom onset. That is the baseline visit for each of these studies. So that may be years into where the disease has already progressed. And a couple of questions here for Christian. Could you please comment on the importance of having a constant clinical input for any and all data science and data analytics steps?
I'm not sure I fully understand what's a clinical input, but of course, at whatever data set you look at, it's important to keep the context in mind and where it comes from. So one question, for example, is that we always ask ourselves is, why do patients perform better in clinical trials than in natural history studies? And a reason could be the motivation to go there, or the way that a patient is actually doing with his disease, or other things. So it's a constant challenge.
And let's see, another question for you, Christian. Could you comment on the challenges with artificial notions like onset of disease in the context of quantifying and characterizing disease progression?
Yes, of course. Age of onset is a very subjective parameter and depends a lot on individual perception. And it's, of course, we use it because it's the best correlating parameter to predict disease progression that we have. But by now we also learned that it works much better on a natural history scale, so on a much broader scale, to distinguish early from late patients. But if you want to distinguish a subgroup that we described in the presentation, like among early onset patients, it's actually not really distinguishing anymore. So that's the challenge with this parameter, but it's still useful to kind of categorize patients into subgroups. Yeah.
Thank you. And here's one I think I can field. Could you please further comment on the importance of confirming the normality assumption for continuous measures before running further analyses? So yes, the normality assumption is often something that's needed for many analyses. And if what's observed prior to performing these analyses is that the distribution is not normal, oftentimes you might need to apply some type of data transformation on the distribution in order to obtain that normal distribution. So for certain modeling methods, such as nonlinear mixed effects modeling models, these assume that the distribution, or the noise, you could say, or the variability on these outcomes is normally distributed. But again, if that's not the case, certain things need to take place prior to executing those analyses. Let's see here, maybe another one for you, Christian. Oh, go ahead.
Okay. Right. Now I was just thinking I could comment on where these measures come from. So that has happened long before my time, but it's true that some of them come from the EMS field and also from Huntington's or scales that were developed in those areas. But that has happened a long time ago. Yeah.
Let's see. Was that you're addressing the question of if we've developed our functional? The last one. Okay. Trying to think. There's a couple of more. Okay, so it looks like we. Go ahead, Christian.
Go ahead, Megan.
Okay, I was going to say it looks like we, I think, have addressed all of these questions, so we can segue right into the panel discussion where there'll be a little bit. You'll hear a little bit more from Christian and I, and be joined by some of our colleagues at FARA, FARA, the FDA, and c-path. Thank you.
I'd like to welcome everyone to our panel. And again, both Megan and Christian, fantastic job. I think you really created a phenomenal impression for what it's like to use the platform to really tackle very specific and detailed questions that are very much relevant to the different rare disease community. Maybe if we could start off with a brief introduction by those of us who are new. So first, Jen, would you introduce yourself just to give folks a context for
Sure, thank you, Jeff. I'm Jennifer Farmer. I'm the Chief Executive Officer for the Friedreich's Ataxia Research Alliance. FARA is the sponsor for the natural history study that Megan and Christian just presented some of the data on. And we also have worked with c-path to put the FA integrated combined database together and reach out to our industry partners who've conducted trials and contribute those data sets. I also, just as a little piece of background, was previously the overall study coordinator working with Dr. Lynch to get the natural history study started. So maybe with that, I'll let Dave introduce himself.
Sure. I'm Dave Lynch from the Children's Hospital of Philadelphia, University of Pennsylvania. I'm a neurologist and neuroscientist here. I've been a PI of this study for the past 19 years, as well as a little bit of a precursor time before that, where we at CHOP follow about 500 people with FA personally.
Thanks, David. And finally, our partner in crime at FDA, Michelle Campbell. Can you please say hi and introduce yourself?
Sure. Thank you, Jeff. My name is Michelle Campbell, and I lead stakeholder engagement and clinical outcomes for the Office of Neuroscience, which is in the Center for Drug Evaluation and Research. But I'm also been the point of contact between the FDA and the RDCA-DAP initiative between the Critical Path Institute and NORD. And so it is always great to be back here for the annual meeting, as it is our third one, as Jeff said very earlier today. But I'd like to quickly thank all of you who have been online all day. And also want to thank all of our patient groups and our patients who have contributed today, either through your videos or being part of panels, as well as your online participation.
Maybe just to kick things off, and then I'll turn it over to you, Megan. What I was really so impressed with is the ability of the platform to kind of get people together and collaborate and use this in a meaningful way, and to draw people like both David and Christian in here who have a lot of historical knowledge about this disease. So even though we focus on the data in the platform, it's really more also about the people and the historical knowledge that also can come together and is essential for moving rare disease cures forward. So maybe David, Christian, from what you've seen, do you think this is an environment that you could see yourself using more commonly?
So I think, Dave speaking, I think this is essentially what we have used as the core of development of everything around clinical research measures for the past 20 years. Everything we talk about clinical trials here, but within this network, matching the clinical trial data to biomarkers, to modifier studies, to just about everything else is done around this core set of information. It has become the dominant aspect. And I would note that in most cases, the CRFs, which we use for the natural history study, are the same ones which we have used for clinical trials, because all the subtleties of expression, subtleties of language for getting the best answer out have already been, in essence, test tried throughout this time. Christian, what would you add?
Yeah, and I just wanted to add it. Of course, a platform like this has many more functions, and not every disease is as well organized as FA is actually. But I think a few other things that some people often overlook is it actually becomes a home for data that otherwise would easily get lost. You could see on the timeline slide that we have been doing studies for 15 or more years, and companies go out of business and the data just disappears. And just providing a platform to host this data that we can continue to work on has a huge value, historically, I would say.
And to build off of that, back to you, Dave. So with this over 15 years of collecting this data, that's quite impressive. So can you touch on how you managed to do that, and over the years, what changes may have been necessary to keep this all going?
So yeah, that's a very interesting question. I mean, the need for such a database began with a clinical trial initiative which started in 1999 for a long since forgotten drug. And then it needed a collection of individuals and needed funding from outside agencies, initially the MDA, followed by FARA, followed by a combination of the MDA and FARA, followed by everything else we've thought of, including the FDA for small portions of it over the course of time. It has undergone some revisions. The initial set of individuals in the room who started this were a group of adult neurologists like myself. Now, I'm different. I practice at a pediatric hospital, so I see kids. But this is a disease of teenagers, and that's the group you need to get with. So there is a conscious effort to recruit younger and younger subjects through the years, to change the selection features as one goes along. The other aspect is we have iterated the measures, but a large number of them have stayed intact or have simply been added on to over the course of time. So you're always thinking of new things to do and how to interpret them without increasing the overall burden. I mean, the place where you have to think about the other burden is not on the investigators, but also on the subjects. They come from a long ways away usually. This is, while I might follow 500 people, probably 50 live in the Philadelphia area. You have to make it easy on them. At the same time they travel to Philadelphia, you want to make it so they're getting a reason to come. Their clinical care, you're getting other questions they have about research in the field asked in a non-confrontational manner. You're educating them about clinical research. And the funding a large part of it goes for partial defrayment of the expenses to come see us. So you have to do all those things to keep people coming back for so long.
So David and Jen, I'm going to bring Jen in too, as you said that you had previously worked on the natural history study at CHOP as a study coordinator. I think, we oftentimes people want to utilize natural history studies, and someone actually asked a question I saw in the chat about, how do we define natural histories? And I quickly looked at the FDA's natural history guidance that simply defines it as, it's an planned observational study to follow a disease over its course. And there were some more descriptions in there and encourage people to look at that guidance. But, sometimes folks, I think, are always trying to develop and trying to capture everything at one time. And I would say that it's important to know what is your end goal of the data you're collecting and what is that research question. And you've highlighted how over time you've had to evolve as things mature, but you must have started off at the beginning, as you said, with just a simple goal of needing to collect data. So do you have advice for folks who are starting out and wanting to begin their natural history data collection in a way that, you know, can be malleable to changes over time?
Sure, I'll try and take the first crack at answering that. When we started, the study was focused on evaluating clinical outcome measures that could be used both for trials as well as understanding natural history. So the scope was not as wide as, you know, understanding the whole natural history of the disease, but around identifying tools that we could use to evaluate the disease. And that nicely parlayed into the natural history study. We started with 6 sites, and we now have more than 15 in 5 different countries. So over time, we've kind of grown the scope, we've grown the target population, the number of sites, and the budget as we could support it. Because, you know, 15 years ago, FARA could not support the budget that we do today. And so there, you know, very logistical things like that I think have naturally evolved over time. But I think it was also important that, at least as an organization, we were counseled by our investigators early on and others in the community that this wasn't a 2 to 3 year study, that this was a long-term commitment, that this was something that we would be doing, you know, until we can go out of business, that this is not something that, you know, will go away. And hopefully it will also be a platform that helps us to really understand the impact of those new treatments as they become approved.
Yeah, and I would agree with that. I think that the advice for people starting out is figure out what the most important question is. Certainly if you're looking to get external funding, because answering questions is what gets you external funding and grants. You have to know what you need to do now, but also have a vision for the future. I mean, the things we talked about, a simple clinical trial measure 20 years ago, which now can be sorted in infinitely different ways, is important, but it's moved on to biomarkers. But you also have to be able to think about the questions which you will answer over 20 years, prospective mortality, things which truly impact that, or things which change the big picture, which is how people really do in life, not simply how they do neurologically. I would note there are budgetary pragmatic issues. For example, early on, we had no cardiac component at all, and we still aren't really able to do a real-time cardiac aspect because it's simply cost prohibitive to pay for echoes and EKGs and MRI scans on every patient who comes in. That just doesn't work. So you have to match your question to what you really need to know short term and think about what you need to know long term.
Thank you for all of that input. So with collaborations like this work, Michelle, how can the FDA play a role in learning from this data and providing comments? And when is it time to incorporate input from the FDA and touch base to get more feedback from the regulatory side?
Right, it's a really great question, and I saw that question pop up a couple times throughout the meeting today. So obviously we are extremely supportive of this initiative and want to see it come to fruition of what it could possibly do, which is help advance and accelerate rare disease drug development. And so as any work in the pre-competitive space, which we extremely do encourage, and what this effort does allow the pooling of the vast data sets and resources for multiple stakeholders. So when is the right time to come and have conversations, such as the example that you've provided with the FARA data, to to the FDA, to the actual review division that may be looking at drugs that are in drug development or in the pipeline right now, and how to get feedback, and how do we begin that connection of taking what's happening to the pre-competitive space and bringing it into the drug development program world on the IND side. So our hope is that when there's an appropriate time and milestone of looking where data has been cultivated and pulled together, and of course that requires enough data to be able to start doing some of these basic, either basic analytics or the advanced analytics that we hope to get. At milestones when data has been generated and there maybe has been some descriptions of what all of the data sets are showing us, we hopefully will be able to operationalize an opportunity to be able to have conversations about and have the data presented to the respective individuals and groups within the center to have a look at this, because obviously we have and want to know as well as everyone else is, what does the data say? What is it helping us? Because it also can help inform us perhaps on the advice we give on making sure we're collecting appropriate outcome assessments or data from our trials, and really help make sure that we're thinking along what the data is saying. So we do hope that there will be the appropriate opportunities at pivotal times when data has been, again, formulated, put together, curated, and something to show for really what could help us, again, looking at what is happening from all of this data pulled together, and how does that impact us in the drug development side.
Michelle, a follow-up question to that, because I know in conversations we've had privately on the team, you and Teresa have both talked about the potential value of the platform for clinical reviewers at FDA, and just by being aware of the availability of good and well-curated clinical data, this is something that could be viewed as a resource and a mechanism to communicate with sponsors, just knowing that the data exists. Could you talk more broadly about the functionality that you'd like to see in the future for clinical reviewers having a resource in that capacity to communicate with sponsors?
Right. And that's a great question, Jeff. And as we continue to evolve this initiative and this platform over time, I think we will hopefully want to continue to increase the applicability and the availability for the different users. And FDA is an end user in this initiative. We want to be able to look at what is happening from curated and standardized data to help inform us. And how can it help leverage, again, individual drug development programs. So there is a hope that's, again, at some point, and I think it will be in the future, again, as we continue to grow this initiative, of being able to, if various clinical reviewers from different offices wanting to see what's going on with data and happening, that will be an option for them. So I think as we continue to evolve the initiative and identifying all of our potential end users, with FDA being one, the ability and capabilities of what that end user can do will help inform. And from our perspective, it can help inform us, perhaps in the advancement and the advice we're able to provide in individual drug development programs, from being able to seeing this curated and standardized data from all of these various data sources, because we often are unable to do that. We only are seeing the individual data that may come in if a trial is actually able to go that far enough where we would see the actual data. So being able to pull all of this data together is actually a real great resource for many stakeholders, including ourselves, and being able to see the entire big picture of what is really happening in the progression of our rare diseases. And really, for example, if we do have had a approved product, how does that now impact a new natural history of a drug, of the course of the disease, and thinking about how we would advance drug development for future indications if we do have something on the market all of a sudden. That's a question that I can see the future where how this could really help inform us down the line when we have more curated data available.
Thank you, Michelle. And to go further into the data usability. So Christian, for you, so from someone who's been devoted lots of effort in analyzing these data sets, from an analytical perspective, what are some of the expectations or comments on the data suitability, and what gaps are there, and how do you go about filling those gaps?
Yeah, the question really is suitable for what? And I think we have made quite some progress in understanding the natural history of the disease in the last couple of years. But now the question really is, how does that translate into the trials that we have? And the gaps there really are most companies that provide us with their data just provide us their baseline data and their placebo group. And I think that is a gap because the context of every individual study is important. So I would really encourage, and it has been discussed also several times during this meeting, to provide all the data from the study, which is not always easy. I understand that there are difficulties with it, but I think that would be an advancement. And another point that I would make is patient-reported outcomes. We can monitor some of those during the natural history study, but again, the behavior in clinical trials is a completely different question. And again, there I can only encourage sponsors to provide us with that data, to learn from it, and to learn how our knowledge from the natural history translates into clinical trials. I think that's the most important one.
Jen, I have
Thank you, Christian.
No, go ahead.
Go ahead, Jeff.
Jen, I was going to ask, you've been contributing data on behalf of patients for quite some time now, and you're sharing it really across multiple organizations, which I think is really a credit to this whole ecosystem. Could you give us some feedback in terms of how the patients see the value in this sharing? We've talked extensively about data sharing, but I think you've done it so well. It'd be good to hear from you in terms of your impression.
Sure. Thanks. Well, with my clinical coordinator hat on, and just being someone who's participated in consenting individuals for these studies, I think 99.9% of the time, people always agree to share their data. It's very rare that individuals who participate in these studies say, no, please don't share my data. So it is something that I believe that FARA, as the sponsor and the organization, continuing to support the study and talking to our industry partners, reiterates that this is the intention of the volunteers of these studies, that they want their data shared, and that we try and honor that as best we can. The other thing that I think we heard from just about every patient today and caregiver is that folks are doing this not only for themselves, but also for their greater community. That they realize that maybe the treatment won't happen for me, but if I can be a part of contributing to the whole that brings us closer to treatments for these rare diseases, that's what motivates people. And that's what, motivates folks to come back for their natural history study visits year after year after year, is if they understand that this is what is essential to move the research forward to accomplish treatments. And, we heard it from, I think, every speaker today. But I think that's, being able to see that opportunity and understand that is what makes these kinds of long-term non-interventional studies successful, or in the case of an interventional study, help us achieve our enrollment goals. The other thing that I know, sometimes I think is important from the natural history study perspective, and one way in which I think patients benefit goes back to something Dave mentioned earlier, is that, understanding the natural history helps us provide better clinical care ultimately. and understanding the disease better and being able to intervene upon it, even if it's not, a treatment for the disease, if it allows us to manage the disease better. And so there have been instances where, what we've learned from some of these non-interventional natural history studies has directly been able to impact long-term clinical outcomes for patients.
Thanks, Jen.
So I think that what you said there, Jen, is really advocating for the inclusion of patients who may be excluded from stricter clinical trial eligibility criteria to have their voice still be heard by participating in these natural history studies. And so that makes me sparks some struggles that I have, I guess, just in whenever we start an analysis. So Christian, I might ask you to contribute here. So whenever you take patients from a clinical trial, why is it important to understand the eligibility criteria when using those patients for analysis?
Should I have a go at this? It's usually one for Dave.
I'm happy to make a comment on it. So, in fact, as you've demonstrated, Christian, and even just a simple cursory look at enough people with FA, the age you're at is a very big predictor of how you're going to change over time, the speed at which you change certain neurologic functions, the type of functions that you change. So when you look at a clinical trial, the first thing you do have to look at is what age was it for? Did they require people to be able to walk? Did they require them to be able to do an exercise test, which is, for example, one recently, which goes pretty closely with people who walk. And once you know that, you can sort of understand, well, what was the predicted rate of change? And if nothing else, for a clinical trial, you can go back to the natural history data and figure out if, well, did the group in the study really change the same rate or similarly, let's say, to people in a natural history study. Those sorts of things really determine who comes in. I mean, there are always other unknown selection criteria. In clinical trials or anything, it's the amount of part. One of them is how far a person has to travel, and how much of their expenses do we reimburse. That determines who comes to do it. And even over time, you realize that the social factors that bring children to come see us are different than the social factors that bring adults to come see us. And that's a key aspect of interpretation of clinical trials, and not so much why they succeed, but in particular why they fail sometimes, because you didn't target the right population.
And I might add to that. I mean, we know that we have a disease of exceptional variability, and every inclusion-exclusion criteria, many of them are designed to reduce that variability. And how to actually do that is not generally agreed upon. So everyone will introduce these kind of small things that we don't necessarily see in the first place from just looking at the data. And then there are many other things that I wasn't aware of at all in the beginning that Dave just mentioned. How much does a patient travel, and what actually motivates this person to participate here? Can make a huge difference.
So I have a comment. Coming from a patient-focused drug development perspective, and we often hear in our rare disease patients of how long it takes for a diagnosis to occur. And in some diseases, because of that, they're delayed the trial opportunity because of inclusion-exclusion criteria. But they still want to be able to contribute in some way to the science. And so I actually would encourage to what Jen is saying, that there is still opportunities for patients who may not meet a trial criteria to be able to contribute data in some way. And then, I think hopefully then our data scientists folks such as Christian and Megan will be able to work through that with the data into what are the data sets that maybe will help inform clinical trial development versus maybe things that would help Dave in clinical practice of what's going on in some way. But I just want to highlight that because we do hear that from some of our rare disease communities where diagnosis can take a while, and then, for example, they may not be able to be eligible for trials due to that. And they're always looking for a way to be able to contribute somehow to their voice, to being able to have their information collected and be useful in some form. And hopefully this will allow that, maybe at the basic analytics level of just the description of the population of what does this rare disease look like. And maybe not in our clinical trial data, but at that basic level of what does, this disease look like in our community.
Michelle, I'm really glad you reiterated that point, because, one of the things that can happen is if we keep getting the same population participating in studies over and over again in a rare disease community, that's not sustainable if you have multiple studies ongoing. And so, ideally we want to understand the natural history and clinical endpoints well enough to have the ability to run trials at different stages of disease. And I think this came up in the Duchenne case study as well, where, they've got now 20 different data sets available from different stages of disease, where you can design trials in, different subsets of the population with the appropriate outcome measures that are the most relevant at that stage of disease. And so, for individuals who might not be included in a clinical trial to come back and participate in the natural history or the biomarker studies, that's what facilitates us to be able to get to the place of having clinical trials in those other subsets, right, of the population, and is, critically important for meeting those needs of studying interventions at all stages of the disease.
Great. Thank you, everybody. And I think we are just about out of time. So I want to thank all the panelists who have participated here today for your valuable input. And we'll now transition over to the next agenda item. Thank you.